First of all, I'm going to start with the acknowledgements. As a microbiologist and someone who's basically on a computer, I'm very grateful for the people we have to maintain our psyllid colonies, as well as growers and industry partners that have provided us with both plant material and diagnostic tools. Funding for this work is originally through plant and food research, and we now have funding through project CRC 202 from the Plant Biosecurity Cooperative Research Centre, as well as support from the Special Crops Research Institute. And part of the CRC project, we have collaborators at Kansas State University, La Trobe University in Melbourne, the State Government in Victoria, New South Wales Primary Industries, as well as Murdoch University in Perth. So we're interested in zebra chip in New Zealand because we have it, but we have just the haplotype A, and we see different symptoms, which I think has been quite well explained this morning. So we see the less dominant striping in our fried potatoes compared to in the US, and as it's been talked about, our infected tubers germinate, which can lead to other sorts of problems. And there's research looking into that, as you've heard, on cultivar, environment, different cultivation techniques, as well as vector behaviour. And what's missing from this, well, we thought, was the analysis of the actual pathogen genome. And so what we're interested in looking at is whether there's a limited genetic diversity of CISO in New Zealand compared to the US, and is that the reason why we see these reduced symptoms? So some history about TPP and CISO in New Zealand. In 2006, TPP was first found in New Zealand, so north of Auckland, shaking hand, down to Taupo, that lake in the middle there. So in New Zealand standards, that's quite a big area. It was found in tomato and pepper glass houses, and the sequencing of the cytochrome oxidase sequence showed that it's a Western US population. A couple of years later, CISO was discovered in New Zealand and named by Leah Lefting. At the same time, there was the identification of Liberibacter in the US as well. But we don't know if the Liberibacter came with the psyllid or not, or if it was a separate incursion event. And as you all know, the current descriptor is the haplotypes. This is, a small part of the genome, and it doesn't represent any sort of biological relevance. We're interested in finding out whether there are biological differences that can explain the symptoms, the differences in symptoms and severity of zebra chip between A and B, as well as between the A's that are in the Americas, as well as what's in New Zealand. And to do that, we've gone with whole genome sequencing. And so this has been done previously for quite a few Liberibacter species, starting in 2009 with an Asiaticus. A couple of years later, of course, is the ZC1 genome. And you can see here on the scaffolds that these early ones were done to completion. So one scaffold means that your genome's been closed. And this is really useful for when you're doing comparative analysis. We've struggled with some of these other data sets down here where they're not complete, they're still in draft format. There's a lot of contigs, and we don't know if all the information's there and how it fits together. So for our analysis, we've used ZC1 as our haplotype B. So ZC1, originally from North Texas, of potatoes, and single psyllid was sequenced. We've sequenced 2 haplotype A samples. The first one, NZ1, from Northland, originally off Tamarillo. And again, a single psyllid was the sample for this. Henny A, which is from Chris. South Texas, originally from potato, and again, single psyllid. As you can see, that a range of sequencing platforms as well as assemblers have been used, but we still have been able to get really good data from all of the three. So to summarise our assemblies, for NZ1 we've got 5 scaffolds, the largest being 0.87 megabase pairs. So this is over 65% of the genome in one contig. And we have sequenced for 3 prophage in this genome. Henny A has 7 scaffolds, the largest 0.69. So again, this is over half of the genome in one contig. And for this one, we have evidence for 2 prophage within the genome, but we haven't been able to elucidate those sequences at the moment. And so with ZC1, here's a comparison. You can see that GC content's all around 35% with 1,200 predicted coding sequences. And there's a slight increase in the size of NZ1, and we think this is mostly due to this extra prophage that we've got. So adding these to the list of genomes, so while we haven't got to completion, we still have a few gaps. They do represent a high quality assembly, and we have been able to get a lot of information from these genomes. So the first thing we looked at was the overall structure of the 3 genomes. So this is a MOVE alignment, and we have CC1, haplotype B at the top as the reference. And what this does is look for blocks of similarity within the genome. And they're nicely color-coded here. And what we're able to see straight away is 3 major structural differences between these genomes. First is the prophage. So these come up as white boxes. There are 2 in ZC1. What you can see is that in these locations, in both of the HAP-As, the prophage aren't there. So the prophage are in different locations in the genome. As I said, we haven't been able to work those out for Henny A. And then for NZ1, we have 3 prophage, again in different regions of the genome. The second one is a genomic rearrangement. So this turquoise block is not in this location in the haplotype A's, so the green and blue blocks join together in both of the haplotype A's, whereas this turquoise block is in both cases on a contig on its own. We haven't been able to work out where in the genome it sits. And the third factor is this inversion. So represented by it being on the other side of the axes in both of the haplotype A's. So this is a block of the genome that's flipped direction. And all of these have been confirmed through PCR as well as our sequence data. So from these, these large structural changes, most of the differences we see are between the A and the B. And so that's the first comparison that we've done. And to look at the differences in more detail, we've gone through and annotated the genomes, so looked for coding sequences. We've used RAST for that, and we've repeated the annotation for ZC1 to ensure consistency between the 3 genomes. As I said, over 1,200 predicted coding sequences in the genomes. We've then used OrthoMCL, which takes those coding sequences from all 3 genomes and clusters them. And it's looking for matches within the genomes through a reciprocal best blast approach. And just a few terms for the next slide. So coding sequence is a region predicted to encode for a protein. Orthologs are coding sequences in different genomes that normally retain the same function and have evolved from a common ancestral coding sequence. Whereas a paralog is gene duplication within one genome. And any coding sequence that's only found in one genome we refer to as a singleton. So the output from the OrthoMCL analysis we can show as a rainbow plot. And we'll just step through what these different blocks mean. So the first one here, most of the coding sequences fall into this core orthologs. So these are coding sequences that are found in all three genomes. So red representing NZ1, green, HENI-A, and blue, the haplotype B, ZC1. We then have these 103 orthologs groups that are in NZ1 and ZC1 but aren't in HENI-A. And these are mostly prophage related. As I said, we don't have those sequences for HENI-A, so they don't show up. The next group is the haplotype A orthologs. This is what we are interested in. So these are ortholog groups that are in both of the haplotype A's. We then have a small number of duplications, so multiple copies of genes within one genome. And then down here, the haplotype B singletons. So these are the coding sequences that are only in the haplotype B. So we've taken, there's 180 haplotype A orthologs, 208 haplotype B singletons. We've done another blast analysis, this time using the whole genome. So this now includes the non-coding sequences as well as the coding. And from that, we've been able to refine our list of unique regions. So the first lot for haplotype B, so this is representation of the ZC1 genome with the two prophage regions, and then triangles showing the unique regions that we've found. So there's 55 of these, and there's one involved in DNA replication and the rest are hypothetical proteins. There's a few clusters here, and these actually are prophage remnants or phage remnants. So these are genes that relate to phage, but there's not enough of them to be a full prophage, and whether this is from excision or imperfect excision of prophage or degradation of old prophage, we're not sure. Similar story for the haplotype A. So in this case, we've represented against the NZ1 genome. The orange tick marks represent the contig breaks, and the 3 prophage again here. So there's 63 unique regions here. Again, a lot of hypotheticals, a few others involved in DNA, modification and methylation, as well as protein transport and some more phage-related genes. And again, these clusters are these phage remnants. So to summarize this, so as I said in haplotype B, 55 coding sequences, 54 of those are hypotheticals, 25 are in phage-related regions, and 31 of them are less than 200 base pairs. The haplotype A is 63 coding sequences, 48 hypothetical, 51 of these are in phage-related regions, and 30 of them are less than 200 base pairs. And this then is where it gets tricky. Working with a sequence that's 200 base pairs means you've got a protein that's around 70 amino acids. It's really difficult to get any more information about these proteins bioinformatically. There aren't conserved domains, they're annotated as hypotheticals in other genomes. And so it's going to take quite a lot more work and possibly more wet lab based in order to understand what these coding sequences are and what sort of role they're playing in the physiology of Liberibacter and how that relates to the symptomology differences that we see. And so another point is, this is based on a limited number of genomes. We don't know if we've captured the diversity of the haplotypes. We've got only one B, we've got one A from the US and one from New Zealand. We could be, so a lot more work needs to be done, and Grant will talk about some of that, and Chris will as well. So just a summary. So we've assembled and annotated 2 haplotype A genomes, NZ1 and Hene-A. We've identified 63 coding sequences unique to haplotype A and 55 unique to haplotype B. They're mostly small hypothetical proteins, and a lot of them are phage-related. We have established a comparative workflow process that we can expand to include more Liberibacter genomes as we get them. But yeah, the ortholog differences that we've identified do need a lot more work to relate that back to the actual biology and symptomology that we see. Thank you.